So I'm Sam, and correct me if I'm wrong, but I think we've all heard of COVID-19. Today I'm going to be talking about the vaccines, in particular the Moderna and Pfizer vaccines, because these are the ones that carry genetic material, and also the lipid nanoparticles that actually deliver them, because we don't inject raw genetic material into our bloodstream. We need to make sure that they're safe and that they get to where they need to be. So my project is sort of split into two halves. I have the half I'm going to be talking about today, which is the self-assembly of these lipid nanoparticles, and in particular, the role cholesterol plays in the self-assembly. But also I take these fully formed lipid nanoparticles and I see how they interact with different cell membranes in the body. For example, endosomal membranes and cancer cell plasma membranes. And this is all sort of under the same arc of understanding and improving the processes of drug delivery design and uptake in particular. So how do you actually do that? Because it's quite an abstract goal. On the far left, you have like a schematic of what a lipid nanoparticle could look like. These are made up of key components like D-lin MC3-DMA, which is a cationic ionizable lipid, which basically means it has a positive charge at low pH. And then there's DSPC, cholesterol, and DMG-PEG 2000. And the last one is only 1.5% component of the overall system. And that's just because it really helps with stability more than anything else. For my project, I'm more interested in the role cholesterol plays because it's known to form domain structures within lipid membranes. And so I've reduced the schematic down to just MC3 and cholesterol in a 3:1 ratio. And then I work at the Airwater interface, which I'll explain more in the next slide. But basically, that means I have to convert this spherical structure into a planar flat surface. And I'll show that in the next slide. But what I wanted to quickly mention is that the genetic material drug that I'm using in the corner here is polyadenylate acid. And this is the poly-A tail of RNA. And this is basically because it's a lot cheaper to get hold of, but it behaves more or less exactly the same. So the Airwater interface is pretty much exactly what it sounds like. The blue in the diagram is water or buffer, which means I can set the pH to whatever I want. And then I just put my flat monolayer on top of this water. And as you can see through the syringe here, I inject the genetic material sample under the monolayer. And this is actually quite new to the ILL. We didn't have this before I arrived. So what you can see in the top right-hand corner is a piece of equipment that I've designed and made myself with the technicians at the ILL. And basically what it means we can do is we can turn on the neutron beam, we can inject our sample, and we can see in real time how long it takes for the genetic material sample to reach the surface. And this tells us a lot about binding kinetics and level of penetration and things like that. So sort of to illustrate this point again, I'm just gonna take an example of surface pressure data. So if you imagine the thin sheet monolayer and you compress it and then you expand it, you can tell in the difference of the pressure of this monolayer a lot about the dynamics of the system. So the main point of this slide is to show that the red data on the right here, this is at low pH. And this has a much, much stronger interaction than it does at physiological pH, pH 7.4 on the left in blue. And this is significant because when you prepare these lipid nanoparticles, you do it at low pH. And then eventually you dialyze them against PBS buffer so that they're safe to enter the body. So these are two very crucial steps in manufacturing process, but they're both bound by very, very different intrinsic properties of the components. So this is some neutron reflectometry data that I took from my last beam time in September on Figaro. In the interest of time, I want you to look at panel D. And this is an example of the kinetic data that I was talking about earlier. So this is where you take the genetic material and you inject it under the surface. And the time axis on the bottom shows you how long it takes to get to the surface. So the dark blue circles are the reference material. This is just pure MC3 physiological pH. And you can see this sort of gradual increase over time. But if you reduce the pH down to 3, in the red circles, you can see it's an immediate interaction. So this means that the poly-A is immediately binding to the surface. But if you go back to physiological pH and then you or you add in cholesterol, as in the light blue circles, you can see that the interaction just is not anywhere near as strong. So here it clearly shows that there is a cholesterol dependence on the interaction, and varying the amount of cholesterol varies the amount of drug binding. So the conclusions I've sort of kind of gone through, but what I really wanted to just reiterate is the impact that this project and research has. So far, it's been really good progress towards fine-tuning the amount of cholesterol that we need to have in these lipid nanoparticles for building and designing these next generational lipid nanoparticle vaccine delivery systems. And with that, thank you for your attention. And for my collaborators, AstraZeneca, University of Manchester, and ILL.